You're at the cutting edge of cardiovascular sciences. I'm your host, Professor John Cook. I'm chair of the Department of Cardiovascular Sciences here at Houston Methodist Hospital. And today we're going to be talking about the good, the bad, and the ugly of oxidative stress, mainly the good, because our visiting professor has done some really nice work showing that oxidative stress is not all bad. In fact, it's important for redox signaling and angiogenesis. And here to introduce our guest is Dr. Li Lai.
Good afternoon, everyone. Today is my great pleasure to introduce our guest speaker, Dr. Uchio Fukai. Dr. Uchio Fukai is a professor and director of the Redox signaling program in Vascular Biology Center at Medical College of Georgia and Augusta University. She got her master and PhD in Kyushu University School of Medicine in Japan, and then she got her postdoctoral training in Emory University School of Medicine, Atlanta, Georgia. Then she became assistant professor and led her own lab in University of Illinois in Chicago. And then she moved back to Emory, where she focused on studying the role of redox signaling in angiogenesis and other cardiovascular and metabolic diseases. And actually, her lab is the first to demonstrate that ROS derived from ADPH oxidase function as a signal molecule to promote VEGF signaling and angiogenesis, as well as post-ischemia revascularization in peripheral artery disease. Welcome to our show, Dr. Uchio Fukai.
Wonderful to have you here, Dr. Uchio Fukai.
Okay. So can I start? So can I start talking my presentation?
Sure. Well, let's talk a little bit first about your background and how you got to where you are now. That might be fun. My understanding is that you got your PhD at Kyushu University. Actually, we were just in Japan. Li Lai and I were in Kyoto, so we're a little bit jet-lagged today. But we had a wonderful visit to your home country and had a wonderful meeting in Kyoto. In Kyushu, you did a very nice work there in vascular biology, and that prepared you well for your postdoc with Wayne Alexander at Emory. Tell us about that, how it was traveling from Japan, where you got your training, to the U.S. to do your postdoc. Tell us about that process, how you made that transition.
Transition from PhD. So by chance, previous postdoc in Cassie Greenring lab, Wayne Alexander's lab, and also another postdoc in David Harrison lab in Emory. So two Japanese postdoc decided to go back to Japan. Then at that time, I and my husband, Toru Fukai, got to PhD. Then we just applied to US, looking for postdoc position. Then therefore, Dr. David Harrison and Dr. Wayne Alexander, Cassie Grindling, kindly accepted our application. Then we had a chance to work as a postdoc in Emory University Division of Cardiology without problem. So therefore, there was no luck after getting a PhD in Japan.
It must be kind of fun to have your partner, your scientific partner, and your husband. You've spent a lot of time together in the laboratory. What do you do when you get outside the laboratory? Do you talk science, or do you have other interests?
Most of the time, we are talking about science, but when we want to relax, we walk along the Savannah River because we are living along the river. So therefore, we walk maybe 30 minutes to forget about the science. And then looking at the beautiful river, and he listens the singing, birds singing. And sometimes we go to the east coast to enjoy the beach weekend. So we really love the the beach. So we don't swim, but walking around the beach is a very relaxing moment for us.
Sounds wonderful. I know that you and your husband were working together in Emory with Wayne Alexander and Kathy Grindling and David Harrison, and you did some of the first work to show that redox signaling, oxidative stress, actually has physiological importance in terms of, I think in the case of your work, vascular hypertrophy. So thickening of the vessel wall is in part due to angiotensin II activation of oxidative stress. I remember those studies. That was quite interesting to learn that oxidative stress could actually have this physiological effect. Are you and your husband, are you continuing to work together on redox and its effects on vascular biology?
Yes. So right now, so I'm shifting to study role of ROS signaling in angiogenesis in endothelial cell. Then my husband started working on role of extracellular SOD and its regulator kappa transport protein involved in cardiovascular disease. So therefore, I'm going to show you today that there is a crosstalk between RS signaling and kappa transport protein, and kappa signaling involved in cardiovascular disease. So we have several R1 multiple PI grants about focus on crosstalk, RS and kappa in vascular disease. So I want to talk a little bit more today.
Great. Well, we're going to get started with your talk, but just to remind those that are listening, you can give us your questions in two different ways. You can go to, if you're online, you can go to polev.com and enter DeBakey, that's DEBAKEY, and respond to the activity. Or if you're on your phone, just text DeBakey to 37607. That number is 37607, and then just text DeBakey and text in your message, and we'll get your questions to our professor so that she can address them. Well, I think, Masuko, why don't you go ahead and get started on your presentation without any further delay.
Okay. So thank you, Dr. John Cook and Dr. Lee Lai for kind invitation to giving me a chance to talk in this webinar. And today I'm going to talk about RS signaling angiogenesis and ischemic vascular disease. And as you can see, RS has a double-edged sword in physiological and pathological state. As you can see, excess RS induce cell death, apoptosis, senescence, and also insufficient RS also reduce proliferation and induce defective phosphodefense. Therefore, excess and low RS are bad. However, physiological level of RS is very important in redox biology, such as signaling function, cell proliferation, migration, differentiation, gene expression, and metabolic regulation. So to keep the RS at proper level, the balance between RS generation and antioxidant defense system is very essential. And their imbalance contribute to various pathologies. As shown, NAD-PH oxidase, NOx, and mitochondria are major source of superoxide. And superoxide is rapidly scavenged by SOD, such as extracellular SOD termed SOD3, and kappa zinc SOD termed SOD1, and mangan SOD in mitochondria termed SOD2. This A2O2 is scavenged by catalase to generate A2O, or A2O2 is also scavenged by peroxyredoxin and glutathione peroxidase. Then A2O2 is very stable, therefore it can function as a redox activator of redox signaling. So here, let me give you my background why I became interested in redox signaling So as Dr. Cook introduced already, so when I joined as a postdoc at Cassie Grindling Lab at Emory, my project was to determine the role of patent FOX subunit of NOX enzyme in angiotensin II-induced vascular growth. And we discovered for the first time that GPCR agonist angiotensin II stimulates HO2 production within 1 minute in vacillus muscle cell, which is inhibited by catalase or by speciation, suggesting that angiotensin II can increase HO2 and which can function as a signaling molecule to promote vascular hypertrophy. And after this finding, we also found that angiotensin II-induced HO2 can activate redox sensitive kinase such as 38 MAP kinase and AKT, which are involved in vascular hypertrophy. So after this finding, after postdoc training, I became more interested in angiogenesis, process of new vessel formation from pre-existing quiescent endothelial cell, which play important role in wound repair and neovascularization required for treatment of ischemic cardiovascular disease. So then I hypothesized that ischemia or its ischemia-derived angiogenic growth factors such as VEGF may be able to generate physiological level of HO2, which may promote angiogenic switch to stimulate endocellular cell migration, proliferation, tube formation, to promote angiogenesis from quiescent endocellular cell. So to demonstrate the role of endocellular-derived HO2 in postnatal angiogenesis in vivo, we use endocellular-specific catalase transgenic mice with Heinrich ischemia PAD model. As shown, endocellular catalase transgenic mice show significant reduction of blood flow recovery as measured by laser Doppler flow and compared to control mice and after femoral artery ligation. Mechanistically, we show that NOx or ECSOD-derived HO2 can promote sprouting and tube formation or activate enos and also promote inflammatory adhesion molecule such as VCAM and MSP1, which promote recruitment of myeloid cell which generate VEGF. Stimulate neovascularization. Our central question is how highly diffusible HO2 signal can activate specific redox signaling to promote angiogenesis. So to answer this question, we focus on compartmentalization of RS signaling as a key mechanism. As shown, we demonstrated that HO2 derived from NOx and its binding partner play important role in VEGFR2 signaling and angiogenesis in endocellular cell. So as you can see, especially using the ES2 hybrid system, we identified IQ-GAP1 as a novel VEGFR2 binding scaffolding protein that coupled NOx cyclic subunit LAC1 with VEGFR2 to promote compartmentalized RS production, which stimulate VEGFR2 phosphorylation to promote endocellular cell migration, proliferation, leading to neovascularization. Furthermore, in addition to IQAP1, we also found that other LAC1 binding protein, ARF6 and P66, also form a complex with IQAP1 to promote compartmentalized HO2 production in caviar lipid rafts. Then promote VEGFR2 autophosphorylation and stimulating angiogenic response. So in addition to caviar lipid rafts, we and others reported that localized RS production at distinct subcellular compartment such as membrane rupture and lamellipodial leading edge and focal complex, where NOx subunit, binding to specific adapter protein, which localize as a specific subcellular compartment, such as wave 1 and IQAP1 and TRAP4, play important role in localized RS production and to stimulate directional end-cell migration. And not only NOx, but also mitochondria, is shown to generate ROS that activate redox signaling. Therefore, we then examine if VEGF stimulation can also increase mitochondria RS in endocellular cell by transfecting cytosolic target and mitochondria targeted redox sensitive biosensor, ROGFP. As you can see, in quiescent endocellular cell transfected is cytosol targeted to ROGFP. The fluorescence signal show blue, indicating cell are in redox state, reduced state. But after stimulation rapidly, cytosolic ROGFP oxidized, then showing yellow and green, showing RS production, which continue more than 1 hour, which is reduced by reducing reagent DTT, and also increased by HO2 positive control. By contrast, when we transfect mitochondria targeted ROGFP, the oxidation is delayed compared to cytosolic targeted ROGFP, and it gradually increase within 10 minutes compared to cytosolic ROGFP, but which continue 1 hour, more than 1 hour, which is reduced by DTT and increased by HO2 and reduced by PEG, mitochondria targeted catalase. Suggesting that VEGF increase cytosolic ROS followed by mitochondria RS production in endocellular cell. Then we show the relationship between NOx4, NOx2, and mitochondria in VEGFR2 signaling and angiogenesis using NOx4 knockdown, NOx2 knockdown, and overexpression of NOx4 and mitochondria targeted catalase overexpression in cultured endocellular cell. We demonstrated that in response to VEGF, and NOx4 derived HO2 activate NOx2 via SERP-dependent tyrosine phosphorylation of RATGET and P47 FOX. And NOx2 derived RS induce serine phosphorylation of P66 and P66 promote mitochondria HO2 production by transferring electron from cytochrome C to generate HO2. This mitochondria HO2 induce oxidative inactivation of PTPRB and which dephosphorylate internalized VEGFR2, thereby increase sustained VEGFR2 signaling, then leading to angiogenesis. We wanted to identify RS target involved in angiogenesis. As shown here, oxidation of reactive system residue of target protein to form sulfenic acid, CSOH termed sulfenylation, is a key initial event in redox signaling. And sulfenylation is very reversible and important for redox relay to form disulfide bond with target protein or forming S-glutathione by reacting with GSH. Importantly, this sulfenylation can be detected using Bauchin-labeled CSOH trapping probe, dimedone analog, which is developed by Dr. Leslie Paul at University of Wake Forest. Using this probe, we performed redox proteomics to identify CSOH formed protein in vivo and in vitro and involved in angiogenesis. So using this approach, we demonstrated redox regulation of quiescent endocellular cell at MAM, mitochondria-associated ER membrane. As shown in quiescent endocellular cell, redox regulatory ER chaperone protein disulfide isomerase PDI acts as a thiol reductase for mitochondria fission protein DRP1 at MAM to maintain normal mitochondria and endocellular function. But so when PDI is downregulated in basal state such as diabetes condition, which induces hyper DRP1 system oxidation, which promote excess mitochondria fragmentation and mitochondria ROS, leading to endocellular senescence. Then, so let me switch to talk about the redox metal, so kappa and angiogenesis. Because we found that kappa transport protein is one of the RS target involved in angiogenesis. So let me start from background of kappa. So kappa is an essential micronutrient, play important role in angiogenesis. Kappa concentration is increased in angiogenic tissue in vivo, and kappa chelator have been used in clinical for treatment of cancer. As you can see, X-ray fluorescence microscopy clearly show accumulation of kappa in wound tissue and addition of kappa chloride in wounding tissue, so facilitate wound closure in skinned wound healing model. However, mechanism by which kappa promotes angiogenesis remain unknown. So since excess kappa is toxic, so intracellular kappa level is tightly controlled by kappa transport protein, such as kappa uptake transporter, CTR1, and kappa chaperone, ATOX1, and CCS, and kappa transporter exporter, ATP7, which deliver kappa to secretory kappa enzyme, such as SOD3 and lysyl oxidase LOX. And then, but when excess kappa accumulate, ATP7 go to plasma membrane to exclude excess kappa. So here, I like to talk about the kappa uptake transporter CTR1 because I I forgot to tell you. So we already published a paper. So endocellular CTR1, endocellular ATOX, endocellular ATP7 play important role in ischemia-induced angiogenesis in vivo using endocellular specific knockout mice. However, only CTR1 is a direct target of VEGF-induced RS. Therefore, I want to focus on, I want to introduce CTR1 in this talk. So what is a CTR1? So CTR1 is a kappa importer and form homopolymer and localize as plasma membrane and increase kappa uptake. However, excess kappa induce CTR1 internalization to protect against excess kappa toxicity. And interestingly, CTR1 has only one cytosolic system 189 residue in the highly conserved C-terminal triad, HCH190 triad, which is required for excess kappa induced CTR internalization. CTR1 knockout mice, global knockout mouse, embryonic lethal. However, agonist-induced CTR1 internalization and its role in VEGF signaling and angiogenesis were entirely unknown. And interestingly, we found that CTR1 is sulfonylated by VEGF-induced ROS at system 189, and which promote disulfide bond with VEGFR2 and facilitate their co-internalization to LAP5 positive early endosome to promote VEGFR2 sustained signaling and leading to angiogenesis. Therefore, oxidized CTR1 is a novel therapeutic target to enhance angiogenesis in ischemic vascular disease, which is recently published in Nature Cell Biology. So in these papers, I just want to show you some key data of this paper showing CTR1 cis1 oxidation is required for postnatal angiogenesis in vivo. As you can see, we developed CRISPR-Cas9 genome edited Redox det CTR1 cis1 alanine knock-in mutant mice. Then using this mutant mice, we show that ear angiogenesis, retina angiogenesis, and skin wound healing mediated angiogenesis, ischemia-induced angiogenesis all are impaired in cysteine redox det, cysteine alanine, cis-CTR1 mutant knock-in mice. In the last part, I'd like to talk about the therapeutic approach to stimulate redox signaling and angiogenesis in disease conditions such as diabetes. As shown, excess RS and insufficient RS impair reparative neovascularization, while normal appropriate level of RS is required for angiogenesis, reparative angiogenesis. So this is called redox window, RS window hypothesis. Initially, developed this model by Dr. Bill Chilian, showing redox window is important for coronary arterial collateral growth. So then based on this idea, we propose that activating redox signaling within RS window is essential for therapeutic strategy in treatment of ischemic cardiovascular disease in healthy and disease conditions such as diabetes. So to stimulate redox signaling in disease conditions such as diabetes, we decided to use exercise because it can restore impaired angiogenesis and cell function in diabetes, which has oxidative stress, by increasing SOD3 that scavenge excess superoxide to generate ATO2 signaling, then reduce superoxide and increase NO. In addition, it is shown that exosome release into plasma mediate beneficial effect of exercise. Then we found that exercise generate exosome in plasma which have SOD3. Then we found that exercise derived plasma exosome containing SOD3 promote angiogenesis by reducing excess superoxide and activating ATO2 signaling in type 2 diabetes. We also show that exosome overexpressing SOD3 can use as an exercise mimetic to promote therapeutic angiogenesis in type 2 DM and other diseases such as vascular aging. Finally, I like to acknowledge to people working in my lab, and also I want to really appreciate Dr. Toru Fukai, who is a long-term partner, working together to focus on role of redox signaling and kappa signaling in cardiovascular disease. And I also acknowledge to all our current lab member and collaborator at Augusta University and UIC colleague, and also Dr. Gus Cho, who is a PDI expert, and Dr. Leslie Paul, who is expert of sulfonylation. And also, I like to acknowledge the previous mentor, Wayne Alexander, Cassie Green. So thank you so much for your attention.
Uchiho Fukai, thank you for that wonderful presentation where we learned about the importance of redox signaling in angiogenesis. And, you know, I think it's incredible how far the field has come in the time that I've been a cardiology doctor, because when I was a junior faculty member, we were all giving antioxidants to our patients with cardiovascular disease because of the Framingham study that suggested that people that were taking more vitamin E had less cardiovascular events. But then when the proper trials, randomized clinical trials of vitamin E and other antioxidants were performed, we didn't see any benefit for patients with cardiovascular disease. And perhaps your work lends some insight to that unexpected finding that antioxidants actually don't seem to help cardiovascular disease, because it's important to have some amount of redox signaling for a healthy vasculature. I'd like to get your response to that and about antioxidants for health generally, because people are still taking antioxidants. But before I do, I just want to remind our audience that you can participate in the conversation now by going to pollev.com if you're online and entering DeBakey. Or if you want to join us by text, simply text DeBakey to 37607. So the number is 37607. Text DeBakey, and then you can send us your question. But Dr. Uchiho Fukai, what about that? What about people who are still using antioxidants for health reasons? What do you think about the use of antioxidant supplements?
So the maybe proposal is, so if we can measure the redox status in plasma, and then then depends on level of redox status in plasma, so we can think about how much antioxidant can maybe we can treat the patient with antioxidants. So depends on the level of redox status.
Yeah, so that makes sense, because you showed us that nice figure where there's a window for redox signaling. A certain amount of oxidative stress seems to be useful. Not enough that's causes impairment. Too much causes impairment. We saw something really similar, right? With cell fate transitions. Cell fate transitions require redox signaling, and if there's not sufficient redox signaling, you don't get cell fate transition. If there's too much redox activation, you don't get cell fate transition. You have to have just the right amount. Goldilocks zone.
Yes. So, and also, as I mentioned, so not only amount, and also location is also very important. So therefore, for example, your previous work, so about NOx4 is important for, some stem cell differentiation, or I forgot, but so.
Yeah, for generation of iPSCs, you need to have some amount of oxidative stress initially.
Right. And then NOx4 is localized at ER or mitochondria and MAM. So therefore, some specific location of NOx4 maybe determines the function. Therefore, maybe if you use NOx1 or NOx2, maybe it doesn't happen.
Mm-hmm. So location is important too. We're finding out that the subcellular organelle location is very important for redox signaling. I think Dr. Li Lai had a question.
Yes, because I'm interested in the self-frenation. It's a post-translation modification, right? And you also show quite nicely that the DRP1 modification is important for angiogenesis. So, have you ever do any other unbiased mass spec screening just to see which protein has been modified the most? And have you studied other candidates?
So we did Yeah, so we did proteomics analysis, so what kind of protein are sulfonylated in response to VEGF, and maybe this may change depending on the concentration of VEGF and also stimulation time. But initial experiment we did 1 hour, 30-minute stimulation of VEGF, focus on signaling target. Then we found that several glycolytic enzymes such as PKM2 and also GAPDH are oxidized, sulfonylated in response to VEGF. So therefore, we are very interested whether RS signaling can modify the endothelial metabolism based on this observation.
That's very interesting.
Earlier you studied the role of angiotensin II on vascular homeostasis and showed that one of the ways in which angiotensin II affects the vasculature is through reactive oxygen species. Maybe just for the folks that are watching, could you tell us a little bit more about how angiotensin II affects the vascular function and vascular structure and the role of reactive oxygen species?
So angiotensin II rapidly stimulate NADPH oxidase within 1 minute, which promote EGF receptor transactivation. Therefore, angiotensin II can activate growth factor signaling through transactivation mediated through reactive oxygen species derived from NOX1. And regarding NOX, vascular muscle cell more NOX1 dependent, and therefore NOX1 also contribute to vascular hypertrophy and hypertension because it has been shown that NOX1 knockout mice and angiotensin II induced vascular remodeling and hypertension are impaired and compared to other NOX. And interestingly, NOX4 case, and there is several paper showing that NOX4 derived 802 is more beneficial, not pathological, which is quite different from NOX1. So therefore, angiotensin II can activate NOX1 to promote vascular remodeling, hypertrophy, and hypertension, but at the same time activate NOX4 to maybe activate some protective signaling to keep the some balance regarding redox signaling. That's also interesting story in angiotensin II.
Mm-hmm. You know, as cardiovascular docs, we think of angiogenesis as being a good thing, but obviously it has also some pathological manifestations. Tumor angiogenesis, even plaque, atherosclerotic plaque, the neovascularization of atherosclerotic plaque contributes to the growth of the plaque. And then age-related macular degeneration with the choroidal neovascularization, that's pathological. So you have a lot of pathological phenomenon that are related to angiogenesis. Are your findings relevant to new therapies for those pathological angiogenesis, the diseases caused by that pathological angiogenesis?
So actually, initially my work focused on reparative angiogenesis in response to ischemia, how NADPH oxidase-derived ROS and maybe mitochondrial RS are important. But in cancer or diabetic retinopathy, still NADPH oxidase contribute to pathological angiogenesis. Therefore, difference is maybe expression level of for example NOX2 and NOX1 are maybe much higher in in cancer and also diabetic condition. So therefore, there is an interesting paper. So for example, NOX2 knockout impair ischemia-induced angiogenesis. However, type 1 diabetic condition, which so impair so ischemia, so reparative angiogenesis condition. Maybe this is due to hyperactivation of NOX2 because NOX2 knockout so rather restore impairment of angiogenesis in type 1 diabetes. So this implicates amount of RS determines the reparative and pathological angiogenesis. But also, so I'm very interested in so little bit I talk about today, so kappa signaling. And there is a crosstalk between so RS and kappa signaling. And then so kappa is really accumulated in so pathological condition, cancer, Alzheimer's disease, and also atherosclerosis. And so therefore, so kappa chelator can so rescue the disease process, maybe due to also inhibiting pathological redox signaling. So therefore, some way, so we want, so in future study, so we want to so more focus on so how kappa signaling and RS signaling crosstalk, then so kappa chelator and antioxidant, so their combination may be very important for treatment of pathological angiogenesis, such as cancer and diabetic retinopathy.
Are you suggesting then that there might be ways to differentiate between pathological angiogenesis and normal angiogenesis for wound healing, for example? You could differentiate between those and then treat one without harming the normal process of angiogenesis?
So for example, in the healing case, so if we use type 1 diabetes, type 2 diabetes model, and clearly angiogenesis is impaired, but this is due to hyperactivation of NADPH oxidase or hyperproduction of ROS, and so also oxidation of DRP1, which we published. Therefore, inhibiting mitochondria fission, which increase hyper-excess mitochondria ROS and contribute to impair wound healing in type 1 and type 2 diabetes. So that's we published in Cell Report.
Well, that's interesting. You know, speaking of diabetes, I was always fascinated by the fact that, as you pointed out, angiogenesis is generally impaired in diabetics, right? Angiogenesis is generally impaired in diabetics. A diabetic person who gets a wound on their leg has trouble healing it. Their angiogenesis is impaired. Yet, they have increased neovascularization in the eye. So that's always puzzled me, that impaired angiogenesis everywhere except in the eye, where it's excessive. Any thoughts about that?
Actually, so that's a so big question. So that so we don't know, but clearly so diabetic retinopathy due to so excess ROS and also mitochondria dysfunction. Therefore, most of the papers showing so treatment of mitochondria inhibiting mitochondria dysfunction and inhibiting mitochondria RS and restore and so impairment diabetic retinopathy. Right. So therefore, some way more, my feeling is more mitochondria contribution, maybe more than so reparative angiogenesis in case of diabetic retinopathy. So, but this is just based on the literature.
Good.
So
therefore, something we have to think about the source. So NADPH oxidase and mitochondria, and so maybe so diabetic retinopathy, more balance, more mitochondria, RS contribute to so retinopathy or mitochondria fission and involved. Something I'm more interested in targeting mitochondria function in this case.
Yeah. Dr. Lai?
So do you think it's possible that in the retina there is a different enzyme? It's like a very special mechanism to produce produce ROS, which leads to different reactions compared with other organs.
Mm. So are you thinking maybe not only NADPH oxidase and mitochondria, there will be another source of ROS?
Yes, different. So you're talking about like NOx dependence. Maybe there are other very, very different source of ROX which contribute to the pathology in the eye.
I see. Okay, yeah. That's we have to identify. Also, maybe the tissue, what kind of cell type contribute to, so ROS also important, because today I was talking about endocellular cell derived ROS, how, so endocellular derived ROS, so from mitochondria and NADPH oxidase contribute to so physiological and pathological angiogenesis. But of course, so macrophage, right? Macrophage, inflammation, maybe T cell derived ROS, and contribute to so lots of so physiological and pathological process. So therefore, we have to study so what's the so cell type so contribute to so RS production. Therefore, so in case of a different disease model, we have to look at whether for example, I have a more macrophage or more T cell, in addition to endocellular cell, maybe pericyte. So what kind of tissue so generate ROS in each so condition? That's maybe you have to study so in future. And also endocellular cell and also macrophage, so cell-cell communication also important. Therefore, so normal condition, there is a balance between so cell communication, but in disease condition, so there is an imbalance of cell-cell communication. So but this communication also create so local microenvironment generating ROS. So that's also maybe important to consider.
So I'm curious about what is your in vivo model? Like what do you use to test your hypothesis mainly?
So right now, so if we focus on so reparative angiogenesis, so we are using hindering ischemia model and skin wound healing model. But so when sometimes so develop physiological case, so we are using the developmental so retina angiogenesis model and sometimes ear angiogenesis model. But in case of pathological angiogenesis, so we sometimes use cancer tumor model and also diabetic retinopathy model, and so also retina ischemia injury model, and of course in so normal condition and diabetic condition. So we are using several so models to study so role of RS in different source and different cell source.
So can you measure the RS level in different cell type in the tissue? Do you have like this technology to just do a sectioning, for example, and just detect ROX level in different cell type? Then you can answer your question, right?
Yes, actually, so right now, so we maybe the best way is so using freshly so prepared so frozen tissue, then adding so dihydroetigene to detect the superoxide production from so tissue. Then we can also we publish the paper so using this DH dye, then co-staining with CD31 to show endocellular cell generate superoxide in angiogenic tissue. So therefore we can do co-staining superoxide in endocellular cell or macrophage and monocyte, something like that, in frozen tissue.
you're talking about the role of other cells in generating oxidative radicals in a tissue. What about the role of other cells in stabilizing the microvasculature? During angiogenesis, of course, vascular smooth muscle cells are important, parasites are important for stabilizing the microvasculature. And I'm just thinking back to your work with back at Emory with Kathy Grindling and the importance of angiotensin II induced ROS in vascular smooth muscle cell proliferation. Does your pathway, does the ROS pathway play a role in parasite proliferation and stabilization of the microvasculature?
So at that time, so we didn't study that area. Only we study the more vascular, vascular remodeling, and by stimulating redox sensitive kinase, and so for example, so phosphorylation of AKT and phosphorylation of EGF receptor, P38 kinase, and which are important for vascular hypertrophy. So therefore, RS always target and kinase, and then induce phosphorylation activation of kinase involved in vascular remodeling and hypertrophy. So that's our data. But after that, more recently, Dr. David Fulton, David Harrison, Dr. David Harrison, working on role of T cell in angiotensin II induced hypertension. So therefore, this indicates that after angiotensin II infusion or generation of angiogenic peptide, then after activation of receptor in smooth muscle cell, and also it also acts maybe also which may recruit T cell and other cell type, then generate more ROS, and in addition to endothelial cell, smooth muscle cell, then that stimulate redox signaling and promoting hypertension. So therefore, definitely, some cell generate RS, but maybe that may trigger to recruit something signal to recruit inflammatory cell, T cell and macrophage, then turn on their signal to create more RS signal to amplify the microenvironment generating RS. That maybe which makes a chronic activation leading to hypertension, vascular remodeling. And regarding also angiogenesis, we published 2000 in Circulation, we clearly show that after Heinrich ischemia and at day 3, a very early phase, and macrophage recruited from bone marrow, then macrophage is a major source of reactive oxygen species, and then which promote later phase of triggering angiogenesis in endothelial cell. Then HO2 generated from macrophage stimulate endothelial cell angiogenesis. Then promote capillary formation. So something we published a paper, time course and the source of RS from macrophage, then followed by endothelial cell and smooth muscle cell, then stabilize vessel formation. So this kind of paper we published.
Wonderful. Well, it's been wonderful to talk to you, Dr. Uchiyo Fukai, about your work. The story of angiogenesis keeps getting more and more interesting, and you certainly have contributed a lot to our understanding of how redox signaling affects vascular biology, and specifically how it facilitates angiogenesis. Thank you so much for joining us on the show tonight.
Thank you so much, Dr. Cook and Dr. Lai.
Thank you
And
very much.
thanks to all for joining us today. You've heard Dr. Uchio Fukai talk about redox signaling and angiogenesis, and so closes our conference today. Thank you again, Dr. Uchio Fukai.
Thank you.